Okay, we'll open this floor for discussion. You've heard three very diverse topics with points of overlap. Who wants to start the bidding? Yeah, Rick.
Rick Myers. Just to emphasize, I was in Group 1, that a lot of our discussion was lots and lots and lots of sequencing being done by many other groups, and how much should, you said it, but how much should NHGRI be doing in terms of sequencing new cohorts or new groups or any type of group, to the point ranging from maybe very little and all the other stuff instead, since I think the phrase was it's going to be generated for free anyway. I mean, you're seeing it from companies, from social media types of things. And so that's controversial, because we range from, how much we should do at NHGRI versus, maybe.
So can I try to understand better what you just said? So was there a consensus view of not more of the same? Is that what you just said, Rick?
Well, Eric and Nancy help here, because I mean it was a big part of the conversation, but it's
so I would say we focused on the things where there was some consensus, and prioritization was not necessarily one of those things, and this comes down to how much is top down, how much is bottom up, and how much will NHGRI drive sequencing for the sake of, looking at more sequence data, and how much will it set standards and create the technologies and software tools to make really complete genomes and so forth. So I think it's not that we don't expect there to be any, and we didn't come to a consensus on the prioritization. I think that's fair to say. But there was discussion of it, for sure, yeah.
Not surprised. Okay, now we're going. Jay, you want to?
I was going to ask you, can you summarize what the competing views were, even if there wasn't consensus?
Oh, I guess we began the session with sort of the following rule of thumb is, what things would not make progress if NHGRI did not take a leadership role? And if the question really was just sequencing, that's it, just sequencing, then I think the group would have consensus that if it was just sequencing, a lot of groups are sequencing. A lot of groups are sequencing in phenotypes. But then it gives you, then you have to much more nuance of sequencing in diverse populations, particularly diverse populations in the United States, number one. Sequencing in individuals with deep proximal phenotypes, methylation, transcription, proteomics, metabolomics. That gives you an essence. So it's not just sequencing, but sequencing with a purpose in a niche, I think would probably be the consensus.
That's helpful. Okay, some hands shot up. Let me try to catch them. Gonzalo, were you trying to?
Yeah, I was just going to chime into this conversation. So I think, I think a little bit like Eric articulated, one of the themes that came up in the big room was that, in the context of just discovering disease genes, there are other efforts going on that might be bigger. There might be things that distinguish an NHGRI effort. And for example, one of the themes that also came up was the data being open and reusable for many purposes. Just after the big discussion, a few of us had a conversation as well. Another thing that's interesting to figure out for NHGRI and for the NIH community at large is, there are huge amounts of genetic data being generated outside. So for example, I think there's an estimate that this year 30 million samples will be genotyped, most of them by ancestry companies, in fact. But if, an interesting thing is figuring out, can we, with an NIH-branded effort, capture some of that data for research, encourage individuals to donate it together with health data. You could probably get a lot more data for your buck by having donations among those 30 million than by generating it from scratch.
Richard, and then
Thanks. Yeah, it's striking to me how the Mendelian program provides so much of the foundation in each of the groups. And as it seems as each of the groups is expressing exceptions to simple Mendelian inheritance that involve interesting mechanisms or interesting genomic changes, there's this layer of discovery that won't be addressed if we take the position that there's so much other sequencing going on out there that simple Mendelian discovery will be further satisfied. And I think we've got to keep that in mind as we think about the portfolio here.
Eric or Nancy, did you guys talk about that at all?
So I think in the context of the bridging, there's a sweet spot, I think, that no other institute is going to pick up. It's relevant for every disease, really. I mean, it's a continuum from the Mendelian to the complex. And so a lot of rare diseases are being funded at other institutes. A lot of the sequencing in rare Mendelian, presumably Mendelian diseases in a variety of institutes, also for common disease. But building the best bridge across that span may be something that NHGRI needs to do because no other institute may have the will or ability to really build the full bridge.
Yeah.
I think the other way not the Mendelian program, so to speak, came up, but Mendelianization came up is, in many ways, if you think about this idea of having deep phenotyping proximal that's been used, in many ways what we're doing is Mendelianize the phenotype. So we're getting a phenotype that's close enough to the gene level that in some respects it's acting almost in a Mendelian way. And so the idea, I think, it's underlying a lot of this. If you're going to understand, gene-environment interaction, it's best to do when you have a smaller number of genes with larger effects and a clear understanding of what the environmental perturbations are. Same thing with epistasis, and the same, again, with structural variation. We're basically looking for models by which we can understand the sort of functionality of genome elements or the mechanism of the association by having phenotypes that are more proximal to the gene level. These are tough things to do with a phenotype such as blood pressure, but having a phenotype that's closer to the gene level, they become tractable problems, number one, where...
Sorry.
She just gave me the hook. I don't blame you. And so the idea is that we can Mendelianize the phenotype to understand the system, and then other groups can take that system or that paradigm and apply it to their own phenotype.
No, I'll use this one. Sorry. No, I just want to say there was a lot of great discussion about particular things that could be done. And penetrance as a concept leaves a lot to be desired. It's really trying to quantify our ignorance about all of the other genetic and non-genetic things that affect a genotype-specific probability of whatever phenome we're looking at. So a rare disease, a particular cellular phenotype, whatever. But it is infinitely preferable to benign, variant of unknown significance, and pathogenic. And the opportunity to use the multi-omics technologies in conjunction with rare variants in patients who do and don't have the Mendelian condition, where we can try to bridge the gap with these kinds of intermediate measures and develop new concepts around penetrance so that instead of quantifying our ignorance, we try to best utilize our knowledge.
Okay, Eric, and then Evan.
Eric Jarvis. So to get at an answer that I see to this question, should NIHGRI be involved into more sequencing? And I say absolutely yes, in the context of the quality, because the current reference supported by NHGRI, all these other institutes are using that. All the other scientists out there are using it. 23andMe and Ancestry and the other companies are using that, which has lots of mistakes in it. All right? And so if you have this, comparison that they're all looking at, I think NIHGRI needs to be in the business of making sure the references that the entire community out there are as complete and error-free as possible. And that does require more sequencing and further development.
It's a clear steer. Evan.
Yeah, I just wanted to add one thing. I think I just realized actually hearing them talk that we missed discussing one thing in the structural variation aspect, and I think it's because we were preoccupied with the technology. And I think one of the questions that we should be thinking about is mutational process. So understanding rates of mutation of different forms of structural variation, sequencing of trios, understanding the patterns of somatic variation in normal individuals. This is something I don't think we don't have a good handle on it in structural variation. One could argue that we don't know enough about it even for single nucleotide variation. But I think this is something that I think NHGRI should take lead in terms of really understanding the pattern of normal mutation and mutational process and mutational bias, because that impacts everything from imputation to association to new disease discovery for missing heritability.
Yeah, I agree. Also, we probably did not do naturally occurring somatic variation justice in the sort of question of penetrance, for example, or expressivity, et cetera. We probably...
When I talk to people in the cancer field, it feels like everything is focused so much on the tumor and tumor, normal tissue, that just a normal pattern of variation as we age, I think, is something that we have the technologies for, but we just don't have really the funding to do it comprehensively.
Right.
I'm just conscious, I haven't looked at this side of the room. Anybody there? Okay. Yeah. Sorry, in the back, I can't see you.
All right. Hi, I'm Megan Dennis from UC Davis. What about resequencing like a cohort like the GTEx or a longitudinal set that we already have a lot of molecular data for, and doing something like doing deep sequencing of the tissue to look for somatic mutations, or doing long read sequencing and being able to connect it to the data that we already have. So like try to integrate, because other institutes or other companies aren't going to go back and rehash what's already been done in these for the cohorts that we've spent a lot of time building.
There is some resequencing being done across tissues in GTEx as part of extended GTEx. I mean, that was one of the projects. The question of how much quality we have, we have the sort of standard quality sequencing as opposed to the platinums that, we'd like to get to, so that there is definitely that. That's an important point. There's a lot of data already in those tissues, a lot of multi-omics data generated in those tissues.
And I'd like to add to that just briefly, because I think it's a great idea. We did talk about with a hidden structural variation going into kind of the existing ENCODE data sets, but it really wasn't up front and center that we should also be looking at generating these high-quality genomes from those same references that we have so much existing functional data from. So I think that's something, too, that dips into our breakout session.
Okay, let's go Stephen and then Katie.
Sure. As one of the few cancer geneticists in this room, I just wanted to come back to Evan and the other important comments about postzygotic mutations, which clearly are being seen in normal tissues. And I think, if we look at what the Sanger is doing is, pivoting towards sequencing normal tissue and trying to characterize the rates by tissue, by stem cells under different conditions and exposures to all the different IARC classified cancer, causing agents. I think it gets at this fundamental question of what can NHGRI do to lead looking at how our genomes fall apart. Mutational rates and mutational, repair, that dynamic is different in every different tissue and the different kinds of exposures, and how we understand that is going to be central not only for cancer, where this is a very pressing issue, but cardiovascular disease, diabetes, neurodegenerative disorders, and the like. And I think how and in what way to structure this so that you can get into all the deep, RNA sequencing, et cetera, to understand the downstream implications of our genomes going awry. Our genomes are not stable, and that's one of the recent things that I think we really are starting to see. And we really need to think about how to do this across all the different disease spectrum.
Yeah.
just wanted to share a point of view from Steven Salzberg, who's not here, but wrote a Bloomberg opinion piece today about genome sequencing and asked the question, like, before we spend any more public money sequencing genomes, can we get clear about how freely available it'll be and how it will actually serve research? And his one argument was that outside of the context of clinical trials might be a place where we could be a little bit less concerned about privacy and experiment with some new ways to let people share their data for the purpose of research. And so that seems squarely in the topic of what we're talking about here and in NHGRI's realm. So I just wanted to share that. It's on Bloomberg Opinions today.
Thanks. Mark.
I just wanted to follow up on, I guess, Eric Jarvis's comment and some other comments. You know, I think one of the contributions of NHGRI that clearly are used throughout the NIH in general is the reference genome. I mean, everyone uses that. And, you know, that's obviously NHGRI's remit. And, you know, one point that was discussed in the structural variation session that I think is very important is that, you know, there's many other institutes that know how to do SNP calling, know how to do genome research sequencing, but there really isn't a lot of people in the other institutes who really understand how to deal with more complicated rearrangements of the genome, how to deal probably with the non-coding genome and all these types of things. And I really think NHGRI has to develop these methods and also really educate people on how to deal with those aspects.
Yeah, thanks. Yeah. I'm not sure we've got your mic.
Sorry. I think this point about Steve Salzberg's article, I think is pretty relevant actually to what we were discussing before regarding the nature of public-private partnership, partnership around genomics and this sort of theme from the first group. I mean, just to briefly summarize one of the views, which wasn't necessarily a consensus view, but it's, by 2030, most genomes are going to be sequenced, and they're not going to have been sequenced by NHGRI. They're going to be sequenced by private companies. So there is a tremendous multiplier effect if NHGRI can help ensure that they're interoperable, that they work well with medical records, and there's actually a countervailing danger, which is that if NHGRI does not lead on this, we could very well be in a situation where the best genetic data sets, the best DNA-seq data sets, are in the hands of private companies who are going to have be able to leverage them for kinds of purposes that I think actually many of us are very concerned about. And so there was this sort of idea of, is there some way NHGRI can engage in that space to make these data sets as interoperable as possible in an ethical way?
In a technical way, or I know you said ethical, but I'm curious to unpack that a little bit.
One of the points was about how like Epic and other health record companies need to be involved in the discussion if we're going to make something like that work.
Want to take another crack?
I just wanted to respond to Arrows's point, which I think is a very good point. One, I think, interesting analogy for NHGRI is kind of like Amazon. Amazon started off selling books, and it kind of cornered the market on selling books. But what it did afterwards is it kind of branched out to selling everything. And it didn't actually sell everything. It kind of made a platform, for how we sell things and how we can do things. And to some degree, you can imagine NHGRI doing that with genome sequencing, not so much doing the sequencing, but providing a kind of platform for genomics for everyone and so forth.
Yeah. Can I ask the panel? So thus far, I think we've mostly been talking about in this session data generation. But how much did your groups, if at all, talk about analysis and tools and methodology versus not just technology side, but that as well, versus data generation and what samples do we want to sequence and gain other information on? Charles or Karen, you want to correct that? And then maybe Andy?
Yeah, so for the structural variation group, absolutely. This is an area where we still need a lot more algorithmic development. We need to be able to integrate the data from different algorithms much more precisely with low FDR rates, et cetera. And then to be able to look at the structural variation across the different types of look at structural variation across the whole genome. I think this is something which needs a lot more development still, and it was recognized by the group.
Andy, you guys kind of alluded, well, you went straight into that, I think, so more than alluded.
So, I mean, the area that we were charged with is G by G and epistate. Those are highly inference-based, and so the demands for going from whatever data we have available to inference, accurate inference of those biological effects is huge. And we didn't get into the weeds much to discuss, well, how do we do this? How do we stimulate it? But it's clear that the demand is there.
I just wanted to add one thing, that was sort of the complexities, the G by E, the epistasis, but one that wasn't discussed was pleiotropy, and I think that's another area where there are huge opportunities because of things like biobanks, where you're getting a large amount of phenomes simultaneously in individuals, and using pleiotropy effectively can give you a much bigger target for discovery. And so I think that's another complexity, that it's not clear that other institutes are going to be as keen to develop as NHGRI might be, because the pleiotropic effects may cut across institutes. And the value comes in really using all of that information, again, to create these bigger targets for discovery. So I think, like epistasis and G by E, the complexity of pleiotropy in the genome, which can be devilish, but also wrapping our arms around it can be a way to enhance the opportunities for discovery.
Yeah, thanks, Nancy. That was explicitly in our comments in the slides. It just didn't make the summary slides.
Well, I wanted to single that general area, sort of the computational biology analysis, not just for specific types of variation or interactions, because I guess back to the point of who's going to do the sequencing. I live in the industrial world, and I think you're absolutely right. I think it'll be done, but at least as of today, I don't think the most innovative and creative novel methodology is being done in that sector. So the data might come out of there. Gonzalo can argue with me; he's never been bashful to do so. But who is going to really invest the time and the energy in those tools to best extract information from that? And it didn't come out in any of the presentations, and maybe it's implicit because NHGRI has always supported that. Wouldn't mind hearing some comments, some thoughts. Yeah.
I mean, I think another issue, just building on what you just said, is one of the points that we discussed was this notion that today most people don't know anything about their genome. 10 years from now, it is likely that most people are going to know a whole bunch of stuff about their genome, and they may be sharing it, with their doctor, but they also may be sharing it on Twitter. And how do we prepare for a world like that? And does NHGRI have a role to play in terms of ensuring, trying to sort of facilitate that private companies aren't, sharing nonsense and aren't creating, hysteria needlessly, and that somehow works reasonably well? And I mean, just in the last few days, these high-profile news stories about how twins are going and having their stuff sequenced by 5 different companies and getting completely different results. Right now, that's possibly mildly amusing, but that's going to lead to catastrophes in the coming years, as lots of increasingly people with less knowledge about genomics are getting little snippets of information and sharing them and acting based on them.
Yeah, Stephen.
Yeah, I mean, I think the computational issue comes back to we've alluded to it through the day, there's sort of the tension between developing things individually and making things available. So in the cancer world, we're putting lots of our genomes together in the Genomic Data Commons in the International Cancer Genome Consortium, and asking investigators to bring the analysis tools to the data as opposed to moving the data, because the data sets are just getting that much bigger that it's harder to manage and errors take place during downloads and manipulations. So where I see NHGRI could have a tremendous effect, I think, is in sort of managing and helping to set those kinds of standards for the different kinds of tools and things as they're developed, so that they're widely available and applicable to different data sets and can be looked at without necessarily having to pull the data to that particular person. And this is something that I think the field is starting to struggle with, but really hasn't really reached the level of having standards and accepted standards among many different groups. And I think that this is a really crucial issue that could make a big difference in accelerating a lot of the questions that were up there, from epistasis to pleiotropy and moving back and forth. And it's the concept of bring the analysis to the data as opposed to how many different ways can I distribute the data.
And you sitting where you do would argue that is an NHGRI focus and not going to be handled by NCI or others, at least in pieces?
Well, I think a number of people are, the NCI is trying to address this with somatic cancer genomes, okay? The GDC doesn't address it with germline and genetic susceptibility, which I happen to be very interested in. But I see, in this discussion, this assemblage of experts, the question is how and in what way to get to the point that the different analytic tools are easily shared. Because right now you see lots of published things and you go get the code and then there are 50 emails back and forth trying to figure out how to get it just right. So maybe it does work and maybe it doesn't work. But that standardization, the way we've sort of figured out most of the linear arrangement of the genome is still not there for a lot of the computational analysis. And that's something that we really need to have if we're going to prevent the people who are in the next 10 years, as I think the comment was just made, of having their own genomes and starting to apply and Google their own tools, and God knows where they're going to get something from the Belarus or whatever that may be corrupt or be doing very strange things. We've had this experience already with some corrupted programs, sent our way. And so I think how and in what way to be really purveyors of the top-notch analytic tools is a critical thing that we have to address. Otherwise, we're going to be washed out. Okay.
Yeah. Let's go down the line here. Yeah.
So this is outside our particular working group, but the idea of tools development resonates, and I think it's something we can all rally behind, just beyond data generation, but leveraging those data by new tool development. But I think what we're hearing is the need to create, for lack of a better word, some sort of an analysis commons where there are sets of data that are well curated, well powered, have, I can go down, have structural variation, have environmental measures, where it could drive novel discovery. But for this conversation, it could be a place where there's novel tool development, hardening, validation, and then ultimately distribution to solve some of the problems that Steve was just talking about and others.
Yep.
Yeah, just to build on that, especially in the structural variation world, there's so many algorithms being developed, and it's really confusing as to which can be used in which situations. I would love to see NHGRI take a lead in educating. For example, let's have a bake-off, for example, a real transparent bake-off of algorithms, and then educate the community, not just NHGRI folks, but others, on here are the algorithms that work well in what situations and what limitations. I don't want to organize that, but I'm just putting it out there. It could be something that could be done. Yeah.
Anyone else want to chime in on that here? From the pen? Andy or Karen? No? Good. Marilyn.
Can I just please remind people, sorry, right here,
hi.
to please identify themselves when they speak?
Marilyn, and then I'll get you this side.
Sure. Marilyn Ritchie, University of Pennsylvania. I wholeheartedly agree with this computational tool area. I think it is really important. I think one of the things that we need to be careful of is that we don't continue down this road of small tweaks to existing tools. And this, I think, is a space where we really could be bold and innovative if we kind of allow for some of the computer science community and data science community to bring some of their tools into our space. I know a lot of people who have tried to write grants to get these new methodologies funded, and I've sat on study section. We're very cautious and nervous about bold, innovative new methods. And so then they go away and they apply their methods to the stock market and to predicting who's going to win the Super Bowl and that kind of thing. And those technologies could be very useful in our space. And so I think if there are ways, maybe through cooperative agreements, where we get some of those people into kind of a U mechanism where they're coming together so that they're not kind of working in a vacuum with these neat methods and making incorrect assumptions about data, but bringing the methodologists with their methods to the data and the data experts, and having them get together, periodically to really try to move those methodologies forward. And I love the idea of a bake-off of some of the existing methods. Bring them all into the cloud, into Anvil, which is, I know, a technology that is being moved forward, but maybe some other cloud, and really compare and contrast and see how they do. I think kind of getting beyond the everybody creates code and puts it in GitHub and then nobody else can use it. I think RFAs that do that we need to get away from, but bringing groups of people with methods together to really be innovative and bold, I think we really need.
Thanks for that. There was a question over on this side.
Yeah, hi, my name is Anshul Kundaje from Stanford. I wanted to say a few things about benchmarks and these kinds of jamborees or challenges. So there have been efforts on this from KG and Dream. I think one central issue is how we increase participation of players in these challenges. One thing is challenge design is very difficult, and it takes a ton of time to design a good challenge. Very few incentives to actually do that, because trying to publish papers and release data is the primary objective versus, say, waiting an entire year for the community to work on some interesting data sets. So I think there are several challenges to actually address challenge design itself and incentivize that. And secondly is to increase participation. So there have been good challenges in the past that have been released, but the participation from key players in the community has been absolutely minimal. Again, the reward structure isn't there. So I think if we are discussing challenges, there needs to be a huge discussion on that on multiple fronts: design, evaluation, incentive, and participation.
Yeah. Thanks for that. I didn't mean to drive this into a computational biology specific discussion, but what I didn't want to do is, it seemed to me like the recommendations around sequencing were heading into not more of the same, but like a whole bunch of different areas. And for me, one of the truisms of genetics and genomics over the past 20 years and NHGR's involvement is this feedback loop of the data generation and the analysis and the productivity. And I didn't want to lose that as we go in. So I just want to make sure we capture that so people know. Yeah.
I just wanted to pick up on the great points by Marilyn. Oh, I'm sorry.
No, go ahead.
I didn't see her, so go ahead and finish, and then Tuli. Yeah.
Okay, so Tuli Laplan in New York Genome Center. So as we are talking about computational tools and being able to use these big data sets, one thing that will be increasingly important as we move forward in large-scale omics data is standards processing, normalization, harmonization, these kinds of approaches. Like genetic data is easy. It's essentially binary data. And even though it has its own complexities, but when we start to talk about large-scale RNA sequencing, proteomics, metabolomics, single-cell data, etc., it's far from a solved question. How do we actually make these data sets accessible, interoperable, easy to analyze? And I think NSGRI could actually have a role in driving those standards and methods forward.
Okay. Did you still want your question?
Yes. Yeah, so this is, I guess, related to all the last 3 points by Marilyn, Tuli, and Anshul, which is that I think we have a mentality right now of having like data archives. Just something, the data goes there and it sort of stays there and kind of calcifies there on some level. And then in case of emergency, or if you really, really want it, then you go through some accessing ritual and download it or something like that. And I think we need to transition much more to the notion of like a data hub. I think there are like literally trivial things that could be changed actually in some cases about how some of these servers work in order to facilitate that, where you could have 100-fold more engagement with those data sets. It would be vastly easier to interact with them. It would be vastly easier to say, hey, when I did a reanalysis in a paper of the data that was in such and such location, it was just following 11 lines of code, and there you can now reproduce it too. And it's really small stuff that could have a huge impact, I think, in that space.
Yeah. Thanks. Yeah, one more on this, and then I want to drive us in a slightly different direction.
Yeah, Adam Philippi, NHGRI. I just want to pile on the discussion about tool development and how it goes hand in hand with sequencing. In particular, I really feel like there's an inflection point happening right now to democratizing sequencing with the advent of the nanopore sequencers, who the company basically gives away like candy and gets into the hands of people sequencing in academic labs, college labs. And this is great because it democratizes the sequencing process, but the analysis component is not yet democratized. And so we have to be very careful in how we fund and prioritize tool development, thinking ahead that this isn't really the top-down structure anymore of the big centers with experts who know what they're doing to everyone with a sequencer in their pocket getting data and understanding what to do with that. And it could be very dangerous if that's not handled correctly. Yeah. And you see a little bit of it in the literature already with many nanopore papers coming out, not all of them very high quality. And so I think that comes down to the tool development, the training aspect, and the standardization aspects.
Yep, thanks. I wouldn't mind putting Eric and Nancy back on the spot. I thought I think your second point was a version of Jay Shinduri's Manhattan Project on steroids. It was annotate all elements, functionally annotate all elements of the genome with respect to evolution, perturbation, natural and engineered variation. I don't know what we left out. Do you want to remind us what the proposal was there and maybe engender some discussion on that? I mean, that's a pretty bold, chunky topic that would seem useful to discuss.
I think the attempt was to synthesize the morning discussion plus the breakout 1, and actually permission across breakout 2 and 3 also brings in structural variation in the environment, and to think about a single project that NHGRI could lead and we would all rally around to frankly annotate every functional element. And by annotating those functional elements, we were including both things that are called genes and non-genic. That's one important part of it. But the other was to bring in this morning's conversation, and now pointing to Jay, having perturbations in a cellular, or COMP2 people are here, I think, in an experimental model system, but then taking that one step further, because of the sheer number of genomes, we have a lot of naturally occurring variation that would be brought to bear. And then I think the shared, and I would say almost novel view of this then is to overlay deep molecular phenotyping, but again, at the cellular level, the experimental organism level, and the human level. And if one brought that together, we would begin to annotate those functional elements, better understand genotype-phenotype relationships, and then ultimately, I said this earlier, develop a paradigm, an analysis paradigm that the other ICs could basically run with in terms of their own disease aspects. And I guess the final thing that I didn't point out is along the way you would need to increase the quality of the genomes across the board. So, have a certain amount of long-read technology, but not just long-read technology. Try to figure out where the holes are and really develop a squared-off data set where you had extremely high-quality genomes, again, at the different cell types, different model organisms, and a relatively large ethnically diverse sample that are well phenotyped at the molecular proximal level. We're abusing those words, but the proximal level, and that would create an enormous resource that I think would drive this field for a long time.
So that's a big one there. I mean, the first advice for Eric Green is you're going to need a bigger budget. Yeah, Daniel.
We all support the bigger budget suggestion. I mean, it is in some ways a crazy ambitious vision, but for the next 10 years of genomics, it seems like exactly the sort of vision that's required. And I like it in the sense that it unifies a lot of what we've discussed, this morning and in various of the breakout sessions as well. It would be, and the other thing I like about it is that it acknowledges the fact that NHGRI will not generate the majority of the data here, but can serve a key role in organising all this genetic data and functional data that's generated across the US and elsewhere, and bring it together in a unified way that is genome-wide and centered around the human genome. And I think that's a tremendous transformative vision.
Yeah, Sharon.
I think to accomplish that mission, it's something that Heidi and I were talking about at one of the breaks. You need to build into that the idea of the human, right? The way you're going to find the humans that have those variants are a lot of people that are being tested, sequenced by testing laboratories, right? So we're also going to need some way to bring in that outside sequencing data that will allow you to have those phenotypes, because otherwise you're never going to have enough humans sequenced by NIH alone. And many of the patients with disease, frankly, who have the phenotypes are being sequenced in genetic testing. So having building somehow into the Manhattan Project some way to facilitate the sharing of data from laboratories is a key part.
And you're referring to proximal molecular phenotypes or clinical phenotypes or both?
No, I was talking about the clinical
Yeah.
phenotypes. The proximal molecular, I assume, is going to be done by CRISPR screens and things like that. But if you actually want to know, does a variant in that enhancer cause a phenotype, or has a variant in that, better put, has a variant in that enhancer been seen in a human? And do we have some phenotypic information? A lot of that information will be in testing laboratories.
Maybe it's because I'm becoming jaded, but it may be more productive not to go to the clinical labs, but to go to the patients, empower the patients to own their genome, and have the patients basically participate and not always going around and trying to beg the participation of those clinical labs.
I would just comment that there are many patients who don't view human genomics as their top priority, who are dealing with serious illnesses or very ill children and would much prefer the labs to provide that information and not be deemed the person responsible. So I think we have to work. Certainly there are many patients who do, but I think we really have to work on both sides of that.
Jay, you were up, but I think Heidi might be useful to bring in right on this thread right here.
Yes, I just wanted to respond. I completely agree with that. We've tried the tact of having individual patients go get their genome sequence from the clinical lab they had it and then share it. It's an enormous challenge for everybody involved. And so I agree that with Sharon that if we could, fund more of an infrastructure to be able to not pay for the genomes, but just have them be shareable by creating a bridge between the clinical laboratories that are generating this data, and even provide them a platform to better capture structured phenotypic data directly from the EHR. The physicians don't want to fill out a paper rec form, and then it goes into oblivion in the clinical labs. So if we could help create that infrastructure, then I think we could capture an amazing amount of information and have a sort of portal or pipeline to take that data in for research that's already being generated and paid for in clinical practice.
Oh, structurally, can I just ask, maybe it's not a question for you, Heidi, but you live in this world more than anybody. Can that be done so it's an NHGRI specific and not owned by the NCIs and NHLBIs and everybody else?
Yeah, I mean, in a lot of ways, my first ClinGen grant, I still have a number of subcontracts to clinical laboratories. Yeah. And in the first phase, we funded them to learn how to submit to ClinVar. And we paid for bioinformatics staff and curator staff to figure that out and do the submission. Now that's considered standard practice, and so we're no longer funding that. But it's a paradigm to think about, a funding opportunity that is just funding the piece that's of interest to us. It's not actually funding clinical testing. So I think there's a model there that, there could be a partnership. Is that what you're getting at?
Yeah, that's what I was wondering. Just, the 1.5% of the overall NIH budget question and bigger. Jay, were you waiting?
So if I could push back a little bit against my own proposal, if I'm allowed to do that. And this is incorporating some live Twitter reaction to my talk, but I think as soon as I use the word function repeatedly without ever really defining it. Yeah. And I think as soon as you start to try to define it, it gets kind of slippery. And so I just, I think in any project like this, and I remain very enthusiastic about it, even expanding it to everything, I think it's important to kind of think through what that definition will be. It probably won't be a simple definition. It'll probably have to be a multi-layered definition and with nuances. But I think having a clear definition and being quantitative about what's been achieved so far, and also what you're going to call success, if you're going to go in that direction, I think is important. But.
Okay, I actually see the multi-layered part of your comment as a positive, not a negative. I think that's actually one of the more exciting things. That definition of function, I think I alluded to this in my question to you, is going to be different at these different layers. And I think that's actually an advantage of this program.
Mm-hmm. Okay, there were some hands. David, were you up? Did you? No. Yeah, Bing.
Bing Ren from UCSD. I could not
believe why I forgot to mention this. I think the word epigenome has not been mentioned many times, and I'd like to mention today, or now. I think this is very relevant to the phenotypes. When we are thinking about phenotypes, we cannot sequence neurological information from blood. We have to think about tissue-specific pathogenesis mechanisms, and that has to do with the epigenome. Why we haven't mentioned this, I think in part because getting tissue is so hard. So we have to find ways to get reliably tissue sources. But also, technologies for mapping epigenomes are expensive. So we need to develop better technologies to make that cheaper, more widely accessible. And third bottleneck is interpreting the epigenome, linking that to phenotype is still biologically a question, right? It's not completely understood. So, but I think we have enough evidence to suggest that epigenome information is extremely useful in guiding us, looking at the functional part of the genome. So I strongly want to add these words to the whatever future plan.
Noted. Yes.
So 2 comments, if I may. One is, language is a dangerous thing. So I want to remind us, we all teacher students, every one of us to a first approximation has one genome that drives 10 to the 13 cells for 100 years. We will never see all the functional elements in our genome. I guarantee you, nobody in this room, I think, will see them. We will measure some. The only thing we see almost all of is the sequence itself. So if you think slightly differently, we would measure some of the sequences in some of the contexts. What we can try to do is build tools that will allow us to generalize our annotations from that. But if we keep thinking or saying even to our students, the young minds that we are forming, we will get them all. I think it's a wonderful job security proposition, but it's not raising the young ones the right way. So that's one comment. The other comment is, I think in general, reductionism is a wonderful thing. Until I understand the molecular phenotype of the gene in one context in a dish, I haven't understood anything. I have colleagues back at Stanford that say, until I understand the hydrogen bomb, I haven't understood anything in biology. I just want to remind you, genomics is the queen of correlations, not of reductionism. You take 7 people that seem to have the same kind of weird disease, you sequence them, and one gene is broken, all of 7 of them. You barely know anything about the gene, but the data says this is the gene. So correlation is queen for us. So just try to temper that with reductionism and with the desire to get all. We will never get all.
I'm going to take that as in pushback if I've just heard incorrectly. You're not against the audaciousness of a plan. You are against hyperbolizing the entire endeavor.
I am against allocating all resources in that direction.
Okay.
Definitely not allocating some.
Yeah.
20 from University of Chicago. I actually want to echo Bin's point using a taxic as example. We all witnessed how powerful, if a method is easy, readily available, how much impact data we can generate. So I was wondering if we can measure active transcription, epigenome, epitranscriptome, or 3D chromosome structure with methods such as a taxic, right? That will fundamentally change the way we collect data. I think that aspect we should also consider.
I thought I missed one over here. Yeah.
So I think part of this activity is you want us to make some concrete proposals to the NHGRI for programs. And I thought it was really striking that both the first and second breakout groups came up with a sort of #1 overlapping priority recommendation, which was to produce highly accurate maps of repeat regions of the genome. And we heard many of those, such as repeat elements have an outsized genetic impact. There's a strong epigenomic impact, hotspot for variation in evolution, and it's a major gap in the finished sequence. In fact, I dare say that we're going to finish synthesizing the yeast genome before you guys finish sequencing the human genome, end to end. Sequencing, sorry. So I'm going to make that prediction. But I have a practical suggestion that perhaps if NHGRI embarks on this program, it could be called the Encyclopedia of Repeated Elements, an encore to ENCODE.
I love it.
I did ask for specific requests and recommendations. Brought that one on.
Encore already exists.
Oh, there we go.
RNA elements.
Oh, right. You got plan B? Yeah.
There's room for more than one.
I just want to make comment on that because I love the idea of having more, and that came up in breakout session 2, is that utilizing ENCODE, making epigenome, but sometimes when you're working with these highly repetitive regions, like I know Evan's been doing a lot of stellar work with the segmental duplications, you have to use long-read sequencing, and that's not something that we have existing data sets for. So beefing up on kind of new technologies to look specifically at these hidden variants or hidden repeat-rich regions could be something outside of just using existing data, but kind of a whole mechanism of how do we do this and make it to where it can extend to the whole genome, kind of like you're pulling everybody through so we can see the pictures of everything. I know that there's been new technologies with Hi-C where people are using long-read sequencing to try to look at repeat regions. RNA's taken a whole new flight with long reads. It seems like there's really an opportunity to do maybe ChIP-seq in a different way where you can start to look at more regional information. And I think all of that could fit really nicely in kind of a new vision of where we want to go to bring everything up to speed with this comprehensive, matching our interest with comprehensive sequencing. And if we're talking about things with ENCORE, I think that one of the most hot spots for these type of things is the centromeres. I mean, the centromeres are absolutely fundamental to our understanding of chromosome biology, yet they're completely missing. The epigenetic information is missing. And I think something like SynCode could be something we work with, because I think that all of the ENCODE data sets were really developed for the euchromatic space. We're missing all the key constitutive centromeric proteins. We're missing some of the constitutive heterochromatin proteins. And there's a whole environment there that actually interacts with the euchromatic space, but we're just missing those components. So I feel like kind of a designated vision there would be useful.
It's actually that's a very good summary of where we've I think we have provided some, in this discussion, sensing a bit of a lull, some direction in terms of where the groups were suggesting to sequence and not to, and the types of technology development, computational biology development, data sharing that is relevant to consider for NHGRI. I don't think anything in a consensus or prescriptive manner came out of this. I didn't expect it to, really. I think the idea probably was to let flowers bloom and give you things to chew on. I think the groups have done that, and the panel's leads have communicated that. Before maybe I draw it to the close, any final questions from the audience, and then I'll give the panel a chance to just bring up anything that we may have missed. Yeah, one question.
So I was really struck by the fact that at no point today have we used the word theory. Genomics has probably the highest ratio of data to theory of probably any of the sciences. There may be really, really good reasons for that, but it's an interesting question to ask of, have we reached a point, are we going to in the next decade reach a point where we have enough of this data that we can start to write down theories, predictive theories of what's supposed to happen in the way that many other branches of science have succeeded.
Yeah. Okay, as I race down the panel, any last remarks? Anyone want the last word? Anybody? Okay, 6 minutes early. We'll give it back to you. Thanks very much for your participation.
Thank you all very much. So we're now going to have a group photo, and that's going to be taken by the stairwell across from the registration desk, I believe, if you come out the door and go right and then left. And I believe Lorgetta is going to be leading the group. And I've been told that people are supposed to take off their name tags for a nicer picture, but please don't lose them. And we will see you all tomorrow. We're going to start at 8 a.m. sharp. So We're giving you the evening free, and please be back here tomorrow morning. It's a different start time, 8 a.m. Thank you.